Welcome to the 10th virtual session, the 20th year of Demystifying Medicine, and today this is also a WALS lecture. The WALS series brings outstanding lecturers whose research has important impact on understanding human disease. In a similar manner, but using a different format, the Demystifying Medicine course aims to bridge exciting developments in medicine with advances in the basic biological and engineering sciences. As illustrated by the Brooklyn Bridge, both WALS and Demystifying Medicine are exercises in bridge building, linking domains which often do not readily communicate with one another. In 1970, the independent discovery of reverse transcriptase in retroviruses, then called RNA tumoriviruses, the work by Baltimore and Howard Temin, revolutionized molecular biology and laid the foundations for retrovirology and cancer biology. Subsequent studies led to the mechanism of retrovirus replication, discovery of oncogenes, molecular cloning, the search for human cancer viruses, and the discovery and treatment of HIV/AIDS. Our speaker today, John Coffin, is a critical participant in retroviral biology and disease research. His HIV/AIDS research reflects his broad interest in molecular biology, virus-host relationships, pathogenesis, and viral evolution and population dynamics. Dr. Coffin graduated from Wesleyan, received his PhD with Nobel Prize winner Howard Temin at the University of Wisconsin, was a postdoctoral fellow with Charles Weissman at the University of Zurich, and since 1975 has been at Tufts University School of Medicine, where he's a professor of molecular biology and microbiology. In 1997, John was recruited to NIH as the founding director of the HIV Drug Resistance Program, later renamed HIV Dynamics and Replication Program. This is in the Cancer Institute, in which he is currently a contractor. Since 1997, he has also been a special advisor to the director of the Center for Cancer Research. Among his many achievements, John is a member of the National Academy of Sciences and recipient of an American Cancer Society professorship. And he continues to advise policy committees at the national level regarding retrovirus-related matters. Let me share two remarkable facts regarding Dr. Coffin, NIH, and Demystifying Medicine. In the 1990s, he and I were colleagues at Tufts Medical School, where, in fact, the idea for Demystifying Medicine was created. By great demand, he has presented exciting research concerning HIV/AIDS in Demystifying Medicine, all but one of its 19 years at NIH and several at Tufts. All told, I've had the pleasure of introducing him annually for almost 30 years. Secondly, since 1997, John has commuted weekly between Boston and Frederick, sharing his expertise between Tufts and the NIH. His Tufts laboratory does important research in retrovirus biology, and his role as advisor at NIH is highly regarded. So the title of Dr. Coffin's lecture today is "These Viruses Are Forever: Consequences of Retroviral DNA Integration to AIDS and Evolution." Dr. Coffin.
Thank you very much, Wynn, for that over-the-top introduction. It has been a very long and very pleasant association that we've had for since the earlier days at Tufts. And I have been pleased to be able to participate in wonderful Demystifying Medicine program for all these many years. To be invited to give also the WALS lecture, I find to be a particular honor as a part-time participant at the National Cancer Institute. I have often seen the invitations to WALS lectures as being given to really the most prominent researchers in all areas of biology, and it's a great honor for me to be invited to do this today. So my talk today going to attempt to cover work that's being done both in my lab in Boston currently and work I'm associated with at the HIV DRP in NCI Frederick. So the first will be on human endogenous retroviruses, particularly a virus called HERV-K HML2 that I will refer to mostly as just HML2 throughout. And I want to particularly discuss the age and distribution and how recently this virus is. These viruses provide us with a fossil record of the association of retroviruses with our ancestors for many, many millions of years in the past, and also whether these have any particular useful functions as residents of our genome today. And in the second part, I will discuss work from the NCI Frederick with colleagues there who've been working intensively on the role of HIV DNA integration in persistence and in the way in which retroviruses or HIV infection is incurable despite the major advances in being able to suppress the virus with current antiretroviral therapy. So I want to start out with a quick introduction to the retrovirus replication cycle. I'm not going to go through the whole thing, but I want to point out in this beautiful picture that was originally made by Eric Fried and modified in very important ways by the work of Vinay Pathak in the DRP, that I want to focus particularly on the step of the integration of viral DNA into the cell DNA. The RNA genome, as everybody knows, is copied by reverse transcriptase into double-stranded molecule of DNA. And that was the major discovery that Wynn referred to earlier. But it's not actually the most characteristic feature of retroviruses. Quite a number of viruses actually replicate in this manner in various ways. What makes retroviruses unique, different from everything else, is that the replication cycle of the virus requires as an obligate step that the viral DNA be integrated into cell DNA. And as this happens, the mechanism is such that these terminal sequences at the end of the viral RNA are duplicated to form this structure called the long terminal repeat, or LTR. And as will become important later on, at the time that it's made, because it's copied from the same sequences in the genome, the LTRs have to have the identical sequence at the time of integration. Integration is mediated by the viral integrase, which comes into the cell and into the nucleus within this capsid structure within which reverse transcriptase occurs. And its integration is widespread in the cell genome, many, many sites. There are tens and tens of millions at least of sites, maybe a great many more possible sites, but it's not completely random. It is directed by certain features. For example, in HIV, integration tends to be into expressed genes, and that's due to its interaction with specific chromatin-associated factors in the cell genome, and the integration step is irreversible. Subsequent replication is via transcription of the integrated viral DNA, and provides a unique mark for the infection event and for the cell in which that integration occurred. And I'll focus on that intensively during the rest of the talk. So the first part of my talk will be work that's being done by students and postdocs in my lab at Tufts University in Boston, shown in the building in this picture here. A nice downtown campus. We're right in the middle of Chinatown, so I refer to it as the best medical school in the country for lunch. And I'm going to talk about our work on HML2, some of our work on HML2. We've been working on this for a long time. First, I want to give a general introduction to endogenous retroviruses. So I view host retrovirus evolution, the host virus evolution follows over eons, follows a certain pathway, and the host retrovirus evolution is a certain modification of that. So imagine a situation where you have a well-adapted virus-host relationship where the virus doesn't replicates enough to be transmitted from individual to individual, but does not replicate to the extent or that the host is killed off by it necessarily. This tends to be the endpoint of the evolutionary process between hosts and viruses, or one of the endpoints. Another is that the virus goes extinct or the host goes extinct. But it's common that you lead to this well-adapted situation. Transfer to a new host usually abrogates that. The genes and the adaptations that make that work in one host don't work in another, and you get a pathogenic interaction. We're undergoing that right now, of course, with the pandemic coronavirus, although that's not a retrovirus. So that leads, of course, to considerable morbidity and mortality. And over a very long period of time, the adaptation, the host can die off, the virus can go extinct, as has happened many times with retroviruses. Or you can reach a benign adaptation as it started with. But in this particular case, and most of the time with retroviruses, it's probably one or the other going extinct. And but what can happen that's unique to retroviruses, almost unique to retroviruses during this period of time, is that you can get the formation of proviruses into the germline, and they can then get inherited. Once they're there, they can be inherited and occasionally fixed in the population as a record, in a sense, of the past virus infection of this particular host. And in some cases, the the endogenous proviruses can express a product which is a dominant inhibitor in some way of the infecting virus, and it can actually contribute to the extinction of the virus infection in the population. So even long after the virus is gone, these endogenous viruses can remain and provide in our germline what's a real fossil record of infections of our distant ancestors going back at least 10 million, 20 million, 30 million years. So, as I said, they provide a fossil record of the ancestral host interaction. And we can study that to learn about pandemics, if you like, that have occurred due to retrovirus infections over and over and over again in our ancestors. So some people like to talk about and you'll see this a lot in reading the literature, talk about the germline as the battlefield between the host and the virus. But I don't think that's the case. I think it's actually the soma that's the germline between the host and the virus. And what the germline provides is the graveyard, is where the losers of these battles end up. And but we can still dig these graves up and read by sequencing the genome and use that to read the past history of these infections. In some cases, in some species, as in mice, the endogenous proviruses, some of them are still alive, can still give rise to virus that can resurrect and cause disease, but that seems not to be true in humans. We don't think, as far as we can tell right now, we haven't had an infectious virus infectious retrovirus of the type that's represented by the endogenous proviruses replicating in us for 300 million years or so, although for about 300,000 years or so. There are, of course, current retroviruses, HIV and HTLV and others, that that do infect humans, but those are very recent introductions. And there may well have been waves of those in the past that did not give rise to a fossil record that we can tell. But this phylogram shows all of the different types of retroviruses. Each blue square here represents an endogenous virus in humans of a different group, and therefore represents a different cross-species transmission or a different wave of infection in our ancestors, giving rise to these that have left behind these endogenous proviruses. There are at least 30 of them. This has happened at least 30 different times, leading to at least 30 distinct retrovirus HERV clades, of which the one of most interest to us right now is the so-called human endogenous retrovirus type K, related to the mouse mammary tumor virus-like, or HERV-K HML2, which I'll refer to as just HML2 from now on. So that it sits down here in this corner, but there are many, many others that we can find. Altogether, about 8% of our genome, about 80,000 different pieces of DNA here and there, many of them quite small fragments, remain in this fossil record. So what do these viruses do when they're in the genome? A lot of them do nothing, as far as we can tell, but others have been shown, for example, to introduce new genes. For example, syncytin, which is a co-opted envelope protein, which plays an important role in placental development. They can also lead to insertional inactivation of genes. Several mutations in mice, for example, and maybe one interesting one in human, has been found to be associated with or to be caused by a provirus integration. They can modify gene expression, for example, by driving the salivary amylase that's found in saliva is driven by a LTR from an old provirus infection. They can form long non-coding RNAs whose function still remains, at least to me, quite mysterious, but they seem to be there doing something useful. In some species like mice, as I said, you can get pathogenesis due to replication of endogenous proviruses. You can get, and they provide, as I've been emphasizing, a fossil record of ancestral host virus infection interaction, and they can protect. What I think is the most important thing they can do is they can protect from infection by related exogenous viruses, and I'll come back to this later on. So depending on how you count, our genomes contain about 80,000 of them. They're all defective for replication. The majority actually are solo LTRs, which are created by recombination after integration. For the most part, their regulation is very tightly regulated by both epigenetic effects and by the transcription factor environment of the particular cell they're in. At the time of an integration into the genome, all but a very few are retroviruses, not transposons. So especially HML2. So the last thing this was, before it was a DNA molecule in the genome, was a virus. And so by looking at it, we can make inferences about what the virus originally was and what it did. As I said, the most recent interdiction of HML2 was about 300,000 years ago. And expression of these elements, particularly HML2, is often seen in normal tissue as well as cancer and various diseases. And we think it likely in those cases that rather than expression causing the cancer, as many people have thought when they've seen this expression, it's actually probably the other way around, that the altered transcriptional environment of the cancer cell causes the expression. But there is also expression in normal tissue as well, which I'll talk about in just a second. Nomenclature of these elements is very messy. If you read the literature, it's really horrendous trying to figure out which is which from one paper to the next. For this reason, we tried to clarify things. Back about 10 years ago, Ravi Subramanian in the lab went through the human genome sequence and extracted all of the HML2 related proviruses that he could find. And from the genome sequence, he could, of course, infer exactly the structure of these proviruses. And as you can see, obviously you can't see the individual ones, but you can see there's a lot of variation in their structure. And we came up with the convention of naming these by the HML2 and then by the chromosome band where they find themselves. And what's important to keep in mind is, again, when you read papers on the subject, you'll find things that just talk about HML2 expression, but that doesn't tell you very much because every one of these has different properties in terms of what it's capable of coding for. Most of these have deletions that prevent them from encoding one or another of the viral proteins. But a useful feature of this is they're all vary at least slightly in sequence, so that if you know the sequence of even a fragment, you can infer what provirus it came from, and from that you can infer what genes it should be capable of expressing. And the provirus in particular I'm going to talk to you about is one called 3Q12.3. It's on chromosome 3, band Q12.3, and it will become apparent later on why I think this one is particularly interesting. So, so what are the functional consequences of expression in normal tissue? What genetic factors are involved in the regulation? And does the pattern of expression reflect the biological properties of the ancestral virus? From this, we can do a little bit of reconstructive paleontology and make a guess as to perhaps what the transmission mode of the virus was from the cell type that it tends to be expressed in, and also perhaps what its pathogenesis was. So we've been analyzing the pattern of expression using the GTX database of RNA expression in autopsy samples. The GTX database is was created by taking autopsy from samples from a very large number of donors. It's up to over 700 donors, over 50 tissues, and we have access to 11,000 RNA-seq samples from all of this. And from that, we can informatically extract the level of expression for each HML2 provirus. So this is a project being currently done by Aidan Byrne and Farrah Roy in my lab. And this just shows a quick overview. Across the bottom, they're hard to read, but are various tissues that are from which we have samples. And this then shows the level of expression of HML2 proviruses overall in a large number of these infected individuals, or not infected, but large number of these sampled individuals. And as you can see, there's particular expression in some brain tissues, as well as the prostate. And we're working on now trying to figure out what the significance of this is. These samples are not obviously diseased, although it has been reported that there is a high HML2 expression in prostate cancer. So we don't know just yet just why some individuals are very elevated in expression here. So the other thing we can do is look at the levels of expression of the various proviruses, again, in all of these different tissues. Here's prostate right here again. And what we find is that the most expressed proviruses vary considerably from one tissue to the next. We think almost certainly reflective of the pattern of transcription factors that are being expressed. The different proviruses have different transcription factor binding sites in the LTR, and this correlates to some extent, at least so far, with what we see for expression. But of particular interest today is this provirus 3Q12.3, which is an exception to that. It's expressed in almost every tissue that we looked at, except in this whole blood sample for some reason. And this actually was particularly interesting to us because we had previously seen this provirus before in another context. And we know that this provirus, from its distribution in species between humans, chimpanzees, and gorillas, it's found at the same integration site in all of these species, which means it must have been present in a common ancestor of humans, chimpanzees, and gorillas. It's not found in older hominoids like orangutans and gibbons, So it's somewhere greater, it's something more than 10 million years old in our genomes and genomes of all of these other species. And it's a defective provirus. It has a deletion in envelope gene that's actually found in a lot of viruses that prevents it from being expressed properly. There's a deletion between the pro and pol reading frames, which causes both of these to be defective, but it has a completely intact GAG gene, unlike its homologs in gorilla and chimp. And it most remarkably has a small sequence duplication in the 5' LTR, but not the 3' LTR, and not in chimpanzees or gorillas, meaning that this duplication must have occurred sometime around the origin of humans, probably. This is fixed in humans. We found in all 2,500 genomes sampled in the 10K in the 1,000 genome sequence database. Every single sequence has this duplication, so it's completely fixed in humans. So these two properties, this intact GAG gene and the fixed mutation here, lead us to suspect that this may have been provided some selective advantage to the individuals that had it initially, allowing it to become completely predominant in the population. This deletion actually, here's this reduplication, right? It is of a short sequence, a 4-base sequence here that creates a binding site. This is the motif for that binding site. It creates a binding site by chance for this HOXPBX, which is a family of transcription factors that are very widespread. And Megan Montesian, who did these experiments originally, found that if you revert that to the wild type, redo the deletion, you lose the ability of this provirus to express a marker gene when it's transfected into cells, strongly suggesting that it's this duplication that created this transcription factor binding site that now allows this provirus to be expressed in all the cells we've looked at. So if you look at this in this phylogenetic scheme, the provirus was originally integrated at this point in the common ancestor of gorillas, chimpanzees, and humans. And the LTR, the GAG gene has been degraded in both gorillas and chimpanzees by different means, but it remains intact in humans and Denisovans, which also have this duplication. And therefore, these events must have occurred somewhere probably early in the human lineage, and we suspect it's really too striking to imagine that this didn't provide some selective advantage for which these people with these proviruses were favored for some reason, and this then swept through the population. So it's an old provirus, just to remind you. Summarize, it differs from the other two hominids in its widespread expression in human tissues. It's preserved the GAG reading frame, and we speculate, and we're now trying to test the idea that the expressed GAG protein may dominantly interfere with virion assembly, possibly leading to extinction of the virus in humans, but not gorillas. A very interesting possibility that we're working to test right now. So the other question I want to discuss is how old are these viruses in us? They were at one time infectious viruses. They've apparently gone extinct. How long was it? So in an effort to identify the likely infectious HML2 proviruses, we developed an approach to search. This was done by actually mostly Zach Williams and Julia Wilchuda in the lab. An informatic approach to search the human 1,000K 1,000 genome sequence project for rare polymorphic proviruses. We found a few, 6 new ones. Polymorphic proviruses are ones that were only found in some individuals and not others, and therefore likely to be the most recently integrated. And we were hoping that among those, we would find ones that were not only recently integrated, but still retained the ability to replicate. We found 6, none of them did, unfortunately. None was infectious. And as far as we can tell, HML2 is extinct, although there could still be some small population somewhere replicating, and was the last turd to go extinct, probably in the order of 300,000 years ago. But what about our closer relatives? We already knew that there were very similar proviruses in many other species. There are some other species. So we used the same approach to look at, we being Zach again, he was the one that actually thought of this, used the same approach with newly released gorilla genome with quite a different result. And so this tree is a neighbor joining phylogenetic tree restricted to the LTRs. And the reason for restricting it to the LTRs is that, as I said, the LTRs at the time of integration must be identical in sequence. And so these trees show both LTRs in these pairs of branches that you can see here, occasional solo LTRs as well. And what you can see is that there is considerable divergence between the 2 LTRs. Since the 2 LTRs are identical when integrated, during the course of evolution, they will drift apart by accumulation of chance mutations and become more and more distant. And the distance between these 2 LTRs then serves as this kind of a clock, not a terribly accurate one, but and nor a terribly sensitive one, since you have to be about 300,000 years before you can begin to see any difference. But a very useful clock for sort of guessing, estimating the time at which integration occurred. And for most of the human proviruses, you can see that there's quite a considerable divergence between the 2 LTRs. If, however, you look at the gorilla proviruses, you can see there are some with somewhat divergent LTRs, but there are many that have all of the ones that say LTR here are pairs of LTRs very closely related. So many gorilla proviruses are much more recently integrated into their genome than are the human ones. And maybe the virus is still active. And possibly, if we could mount an expedition to Africa to look at some gorillas, we could find some still replicating virus of this group. I want to also point out one other thing. If you look at the scale bar on this bar here, which is not much different from the total length of this tree from the common root, is about 2%. 2% is like the divergence between 2 HIV sequences of 2 different people on the same transmission chain. So this is 10 million years, and HIV, you can get this kind of divergence in a year or 2. And so these are really amazingly well preserved sequences that have had very little replication as a virus since the time that they were first integrated. So I'm going to move from that now to talk about work that was done by my colleagues largely. They did all the work. I did much of the analysis that I'll show you, but they generated the data that underlies it. And we'll talk about characterization of HIV persistence. The current antiretroviral therapies fully suppress HIV replication. As far as we can tell, there's absolutely no replication going on, although this point is still a little bit controversial. And it's one of the medical miracles, if you like, of the last century. They allow infected people who had originally had a death sentence of HIV infection to live a mostly normal life. But there's still both individual and, of course, global challenges. Only about half the affected population has access to drugs, for example. Effective means of blocking transmission with these drugs are slowly coming into wide use, but a vaccine is still a remote dream as far as I can tell. Development of resistance antiviral drugs is not much of an issue in the U.S., where we have a large supply of drugs, a cabinet full of drugs to fall back on, but it is in poorly resourced areas where the choices for drugs are very limited. But the most important thing I want to point out is that antiretroviral drugs can effectively suppress HIV infection, but they will never cure it. And the reason for that is that the drugs block the replication steps in HIV, but once a provirus is inserted in the cell, they have no effect whatsoever on that cell or on the provirus it contains. And so whatever that cell can do, it can survive the virus. So why can't we cure the infection? Well, the reason is that the virus is in a type of cell which is designed to, in a sense, live forever. In a CD4 positive T cell, key component of the immune system, which can respond to antigen and then go into a resting state that lasts essentially forever, and can only, but ready to be restimulated if the antigen reappears. So what antiretroviral drugs do, if you look at HIV infection, the course of HIV infection, there's a steady state set up that varies from one individual to another over a very wide range. And this is a state of constant replication of the virus. This is marked by studying plasma HIV RNA, measuring the amount of viruses in blood, essentially. Antiretroviral therapy stops replication in its tracks, and the virus in blood declines very quickly and reaches usually a very, very low, but not zero level that can last, maybe decays very, very slowly, but can last truly for many years. If you look at viral DNA in cells, you look at the provirus in cells, you find that drops fairly quickly, but only about tenfold, and then levels off. So there remain many, many cells, probably 10 million to a billion, somewhere between 10, 100 million and a billion cells still in the body somewhere that have HIV-1 DNA. It's been shown by others that most of this DNA is highly defective, but a fraction of it is always capable of making infectious virus. It doesn't do so most of the time. This little residual viremia comes from the occasional one of these cells that for some reason is induced to make virus and release some virus, some virions into the blood. In all patients, virtually all patients, very few exceptions, if you interrupt therapy, then the viremia returns to the level it was, and the course of infection simply picks up where it left off. And to date, we have there's a very active research on this. I'm not going to talk about it, but there is no known way to prevent infection except by the extreme measure of whole body ablation and bone marrow transplant that Timothy Brown, the so-called Berlin patient, had many years ago. That completely eliminated and he was transplanted with cells that were genetically resistant to HIV and survived the transplantation drug-free. Unfortunately, he died last year of the recurrence of the cancer that he was treated for originally. But he nonetheless provides sort of the model for the potential for HIV cure. And there have been two other patients I know of who've had the same outcome over the years, but it's not a treatment that unfortunately you can do for everybody. So how is this HIV infection persisting? Well, there have been two general ideas. One is that there's persistent low-level viral replication in sanctuary sites in the body that the drug is not reaching effectively, or that it's due to non-expressed latent proviruses in infected cells with the complete absence of replication. And our evidence strongly favors the second possibility. And the way we can get at that is to take advantage of the fact that HIV replication is slightly error prone, as it is for all RNA viruses, and that in an infected individual, untreated, HIV the infection is sustained by over and over replication of the virus, and therefore continuing ongoing generation of viral diversity. Whereas if once the provirus is in a cell, if that cell is dividing and making the virus that's being made will be genetically homogeneous, and you will get you will see in both in the viral cell's DNA and in the virus that comes out, rather than a highly diverse population where every single sequence you get is different. You get these large what are called rakes of sequence, well, where you get lots and lots of identical sequences because they're coming from these expanded cells. This is an analysis that was done for a paper we published a couple of years ago, done by Mary Kearney. These are patients that were treated very early in infection, just within a couple of months following their infection. And the open symbols indicate the viral RNA in these patients at the time that just immediately before therapy. And then the closed triangles here indicate the sequences 3 years or so later, where you would have seen in a patient that was replicating a virus, if there's any virus replication, you would have seen the kind of genetic diversity that you saw in the last slide. And instead, there's no diversity whatsoever. All of these have basically 0 or 1 or 2 nucleotide change, the same difference from the start, the same as to the same extent as the pre-therapy samples. There's absolutely no replication, indication of any replication in these patients over this period of time. So to pursue this further, we're interested in looking now at what determines where where integration sites are, what you see in these patients in terms of integration, and what is the effect of these integration on the cell itself? How does that contribute to the persistence? So HIV can, as I said, can irreversibly integrate in millions and millions of possible sites in the cell genome. And each site of integration then can uniquely tag a single infected cell and all of its progeny. Okay. In long-lived HIV-infected cells, sites of integration are determined both by the initial preferences, which we can refer to as hotspots, and I'll show you what that looks like in a second, or by selection, as modified by selection after integration, or as modified by chance outgrowth of clones of cells directed by other means, such as the immune response. We use an assay that was originally developed by Rick Bushman and Charles Bangham, which involves shearing of cell DNA, ligating linkers, PCR amplifying, and paired end Illumina deep sequencing, so that we could get both the sequence of the shear point, the break point at which it was sheared, as well as the integration site, the junction with the LTR. The integration site then, from the LTR primer, you read the integration site, and from the breakpoint primer, from the linker-specific primer, you read the breakpoint. Multiple sequences with identical integration sites, but different breakpoints, imply clonal expansion of the cell after an infection. That's an important concept here. That is to say that you can, from a single DNA molecule, you can only get one breakpoint, no matter how many amplicons you get out of the Illumina. So if you see 2 breakpoints, there must have been 2 DNA molecules, and those can only have come from 2 different cells. So the number of breakpoints you get in this is a measure of the number of cells you start with. This is just a cartoon visualization of the process. You extract the DNA, you shear the DNA at various breakpoints. You add a known linker sequence to it. You PCR amplify between linkers I'm sorry, but primers recognizing the integration site, the end of the LTR, and primers recognizing the linker at the breakpoint. And then from these paired end sequencing from each end, you can get both the integration, determine both the integration site and the extent to which that particular the particular originally infected cell was amplified to give a clone of cells. And so so going backwards, sorry. So first we just compared the integration sites sort of globally, depending on the nature of the gene that they were in. So we looked at genes, so we sorted out genes by measuring the level of expression using RNA-seq analysis, and then we get the TPM. And then we binned, out of 20,000 genes or so, these were then divided into 100 bins of about 200 genes each, separated, ranked by expression, level of expression. And so we get this nice green curve here. And then we looked at the numbers of integration sites in each of these bins, which we actually lumped together because it's otherwise be very noisy. We lumped these together just into 4 bins. So actually 5, not the ones that were not in genes, not expressed, you were at a very low level. And then as you can see with increasing gene expression, you get more and more integration sites. This is actually shown, this is not new, it was shown by Rick Bushman many years ago that HIV expression in PBMCs favors in peripheral blood mononuclear cells that it's particularly in CD4 cells in that population, those favor gene expression in a very strong way. And that is a important property of HIV. If we look at the samples taken pre-therapy before we started or on therapy, we can see also this very strong relationship, but we can see that actually with increasing gene expression, there was less and less integration into these integrants found in these 2 sets than we found in the in PBMCs. So to look at this a little bit more, we just took the ratio of the PBMC integration, the ratio of this number to that number or that number, and looked at that. As you can see, the ratios, it's really showing you the same thing, that this ratio increases with increasing gene expression, and it's quite low in non-expressed genes. The higher the ratio now, the greater the relative selection against the cells that have the integrated proviruses. So this clearly shows there must be selection against cells with integrated proviruses. And that selection is stronger, much stronger for cells with in the same orientation as the gene, as opposed to the opposite orientation. That makes sense because a provirus, the same orientation as the gene, will insert polyA sites, it'll insert splice sites, it'll insert all kinds of things that will disrupt the transcription as the RNA polymerase tries to make its way through it. But there are so that's what the first property of that can alter the original distribution of integration sites in during the course of infection and during the course of therapy. So let's look at the distribution of integration sites. This now is these are done using an Excel-based mapping program that I developed that takes all of the integration sites in a given region of the genome, or takes a given region of the genomes that you specify, divides it into 250 bins, and then counts the number of integration sites, either in the same orientation as the genome in red, or in the opposite orientation in blue, and plots those. and plots those along with the level of expression of the gene or genes that are in the same bin. And what you can immediately see again here is a very nice correlation between the level of expression of genes and in the frequency of integration this is now over the whole chromosome. This is all of chromosome 16. This is about something like 20,000 integration sites all run together here. And and this is in PBMCs that are infected in cell culture, therefore in probably all in CD4 cells. And this is in a pool patient samples derived from a number of DRP studies. And what you can see is there's a very, very strong similarity between what the pattern in cell culture, the pattern you get in infected individuals. All of these are unique integration sites. We're not showing multiple breakpoints in any of the figures I'm going to show you for a bit here. But what you can see is there's one place right here, one bin that has a much higher level of expression and also a much stronger preference for integration in the same orientation as the chromosome than in the opposite orientation of the chromosome. So let's take what's in this box and expand that. And now we can see again, this is now just a 10 kb stretch of the chromosome. We can see again, you have this very strong preference. Integrations tend not to occur in regions, tend to occur only very, very rarely in regions that are not within expressed genes. And and are all virtually identical in pattern between the PBMCs and these pooled patient samples. These are all patients that have been on therapy for a while. But again, you can see this very strong hotspot right here. And we'll zoom in still more. Again, you can see the hotspot. Now you can see the name of the gene appearing, MKL2. And now we focus in just on the MKL2 gene. Now what this program does is it breaks the gene up into introns and exons. If you specify exactly a whole gene, it breaks it up this way. And so you can see that all of these genes, this high peak of integrations is a cluster of integration sites in this very short region, 3 exons, comprising the introns between exons 4, 6, and 7, and multiple integration sites all in the same orientation. And we can zoom up. We can look at this even more closely by oops. Oops, we will do that later. And so this is a very small region. This is about 7 kb, this whole region, smaller than the provirus itself, actually. And this is a very, very striking, highly significant concentration of proviruses here. Clearly has some meaning. This very much resembles the kind of thing that one had seen, retrovirologists had seen many years before in cancer cells. These are not cancer cells. These are cells that have been clearly expanded, and they've clearly been expanded to some extent because of the presence of these proviruses. And what proviruses can do is they can modify the LTR, can drive expression of a nearby gene as well as the expression of the provirus. And so this shows 3 genes, the 3 genes that were originally identified. There are a few more that I'll show you in a minute. That and the location of the integrations with respect to these genes. In BOC2 and STAT5B, the integrations probably drive expression of the whole gene. In MKL2, they drive expression only of a fragment of the gene, removing what is probably a negative regulator from the rest of this. MKL2s, these are all genes that are involved somehow in regulation of expression and regulation of cell growth. MKL2 is involved in the serum, is a gene involved in the serum response cell of cells. And as you can see, it will drive a fragment, it will drive expression of a fragment of the gene downstream fragment, but not without this part. And these kinds of effects have been reported many years ago in retroviral-induced cancers, but I want to emphasize these are not cancer cells. We have also seen integrations in STAT3 driving expression, not in patients, but in vitro infected PBMCs that are passage for a while. Experiment done by John Yoon in my lab a couple of years ago. So in some way, these proviruses must act by stimulating growth or interfering with apoptosis of these genes. We actually don't know which, but they could they could become emphasized this way, in either stimulating growth or affecting survival of the infected cell. Since then, we've identified a few more genes in a much more recent study that I've been working on with Steve Hughes. I think it's about to be published, we think. And that will these are less apparent. You need a larger database to see them, but they still have quite a strong level of statistical significance for finding the integrations being enriched in these genes or having a strong orientation bias in these genes. That may be about all there are. Of all of the other genes we've looked at, the frequency that might possibly be involved, the frequency of integration of PBMCs is quite low, and they're probably unlikely to get enough integrations to cause this to happen. So this is the zoom in that I promised you before. This just shows now exons 4 through 7 and all of the integration sites. And this is in a 7 kb region out of about there are about 30 integration sites out of a few thousand, out of 30,000 altogether, which is a large fraction, considering this is a tiny fraction of the 3 kb genome. So now all of the things I've showed you before are unique integration sites. We've eliminated all of the breakpoints, reduced them to 1 for each individual gene. Now, if we restore this and look at the breakpoints, what we can see is that some of these sites, if you look at the scale here, this is now, one of these was amplified, so it was seen 50 times more than more than a cell which gave rise to a single one with a single breakpoint. And so you can see this clonal amplification quite dramatically here with some of these. If you look at the scale on the x-axis. So, and that means that the cell with this integration site has divided a lot. And so what's all about with this clonal amplification? Well, if you look at, this is now the original individual we studied, refer to him as patient 1, the one that had strong integrations in MKL2 and BOC2 and the other STAT5B and some of the others. And if you look at the distribution of integration sites by the number of breakpoints that you see, you can see that about half of the integration sites that we saw, half of the integrations we saw were in sites with that gave rise to more than 1 breakpoint. In other words, that had been clonally amplified since this patient was infected, and that was and since treatment had started, and this patient had been infected for about 20 years, treated for about 10, and these are integrations. So the gray is integration sites identified only once. But identified only once doesn't mean they were not clonally amplified. We can only see about 1 cell. We can't sample the whole patient, of course. We can only see about 1 cell at 10 to the 5th or 10 to the 5th to 10 to the 6th in that patient. So integration sites that are identified only once may well also all be in clones. In fact, we think they are. We also spotted a very interesting one in a site that was not in a gene was a little bit ambiguous. It was repeated a few times. It was found in more than 10 million cells, we calculate, over the whole body. And this provirus gives rise to infectious virus that is actually remains, it was detectable in plasma as low level viremia. So a few of these cells, most of the cells are not expressing RNA, but a few of them from time to time are induced in some way, we don't know why, to express RNA to make virus, probably die of it. This gives rise to a detectable viremia, and it gave rise to a detectable viremia in this patient. So are all the HIV, we think in patients on expanded clones, we actually think so, but we can only sample about 10 to the -5 of the CD4 positive cell population. Some of my colleagues in Frederick, Steve Hughes, is working on, and in collaboration with John Mellors, the University of Pittsburgh, is working on a project involving analysis of very large numbers of integration sites from very large sampling in patients and trying to get a handle on just what fraction of the population really is in clones. But it's a very difficult problem to figure out without being able to see all of them. And that's, you consider the problem of looking at the city of Boston in the fog, all you see is the tip of the tallest building. How can you infer from that what the rest of the city looks like? Well, that's what it looks like, but I don't know how you would have guessed that otherwise. And so it's going to be quite unclear to figure out it's going to be quite a difficult project to figure out just what the distribution of integration sites is from the relatively small sample you can do. So clonal expansion of infected cells, you think, can arise in several ways. First is direct effects of integrated provirus on cell multiplication and survival. The second would be chance outgrowth of memory cell during homeostatic replacement, which is a property of course, immune cells. And the third is antigen-driven expansion during active immune response. And actually, we know from the work of a former DRP member, Francesco Simonetti, published a very nice paper last year showing this process did occur and came up with a clever way to identify just what the antigen was that was being recognized by the infected cell. So this is clearly relevant to this process. It may be the most important. The latter two effects have nothing to do with the proviruses, but the provirus serves to mark all of the dependents of an originally infected cell. So you can follow that and see what's happening. We can learn something. So we can learn something about this if we look at the relationship of amplification ratio to the type of integration site. In other words, for integrants in different types of genes in different regions, what is the amplification ratio? Basically the number of breakpoints per integration site. And so if we sum that over all sites for both pre-therapy and on therapy, pre-therapy we get a bit of amplification, about 20%, 1.2 amplification ratio, goes up to about 1.6 in all sites on therapy. It's exactly the same. Maybe a very, very slight, but certainly non-significant blip up in highly expressed genes, but it's pretty much the same throughout in both pre-therapy and on therapy. On therapy, of course, is a mixture of latently infected cells as well as actively replication cells, and then if you actively replicating cells, and then if you look at all of the genes we identified, we cannot see much more amplification in those. Overall, maybe a little more, but probably not significant given the numbers of amplification directly due to the effects on these genes. So we think this is actually more consistent. This might be more suggestive that these genes are, an integration in some of these is affecting survival of cells rather than driving amplification of cells. But that remains to be seen. And I will summarize. So HIV persists during suppressive arc by survival of infected cells with non-expressed provirus. It's modified by selection against cells in the provirus expressed genes, particularly those in the same orientation. Now, could that be the effect of provirus on gene expression? In other words, if the gene is, if you knock out expression of one of these particularly highly expressed gene, is that uh, causing is that causing the cells to be less viable, or is the fact that the cell is as others have proposed, the fact that the cell is in a highly expressed gene, mean that provirus tends to be more expressed and therefore more damaging to the cell? And that really, we favor this alternative, but that really remains to be seen. There's selection of genes, cells with provirus in a particular region of a few specific genes, which could be stimulating division or promoting survival, or perhaps both. And clonal amplification is largely independent of the provirus integration site. It's mostly likely just marking immune-stimulated cell proliferation, and it's not a global effect that is really driving persistence or expansion of infected cells. And so I'll close with this and thank lots of members of my lab over the years who worked on these projects. And I will thank a great many members of the HIV DRP and collaborators at the University of Pittsburgh or in Leidos Biomedical for all of their help and providing really all of the data that I showed you in the HIV project. And I will of course thank all of the patients, all of the participants in this project who donated samples to it. And I will close with that and look forward to your questions.
Thank you very much, John, for really very exciting presentation. We have a fair number of questions that have been asked. Let me begin with Stefan. He says, is it possible to get complementation via reassortment? For example, is it possible to get GAG from one provirus, envelope from another, etc., in order to get an infectious retroviral particle?
Yeah, yes, the answer is yes, that can of course happen. It's a little bit complicated by the fact that GAG and Pro and Pol are made as one precursor, and it's the precursor that's incorporated. So you can't really get complementation between individual proteins within the precursor, but between Pol and Env, you can certainly get complementation. It's a very important part of retrovirus biology.
Okay, Gerald wants to know, have analyses been done to suggest whether any of the integrated viruses seem likely to have generated adaptive traits due to influence on genes relevant?
I'm sorry, can you say that again, Wynn?
He says, have analyses been done to suggest whether any of the integrated viruses seem likely to have generated adaptive traits due to influence on genes relevant?
Yes, I think that's certainly likely to be true. And there's been quite a lot of speculation on that, and there are a few concrete individuals. I cited the example of salivary amylase, which is a pseudogene, originally a pseudogene of the pancreatic amylase, driven by a salivary gland specific expression of the endogenous proviral LTR. And there are probably a lot of other examples of that. There are examples that people have suggested, but it can be sometimes rather difficult to test.
So Mark wonders whether retroviruses are more susceptible to mutations because the single strand is less protected in the process.
Well, there's something to that. They're more susceptible to mutation than cell genomes or most DNA viruses because the enzymatic activity it lacks a proofreading function. Now, we can speculate that actually is because of they're also more susceptible to environmental damage of that sort. Um, particularly, a particularly common mutation is chemically or otherwise induced is C to U change. That occurs spontaneously at a very low rate. It also occurs by the action of cellular deaminases as well. And it's likely that the reason that RNA genome viruses, most RNA genome viruses, actually coronavirus is an exception, don't have a proofreading activity is there's no point to it if there already is a high background error rate, mutation rate due to environmentally induced mutations.
So Henry asks whether there are polymorphisms of HML2 in human, with some alleles being, for example, solo LTRs and others containing GAG or separate than in the HML2 itself.
Yeah, Henry, I'll send you a reprint on that. We published a paper on exactly that about 15 years ago, maybe 10 years ago. I forget exactly, maybe more like 10. Which is true. Yes, there are a number of polymorphic proviruses like that, and there are some cases where you can see different individuals where the recombination to give a solo LTR has occurred independently. You can tell that because there's slight differences in sequence between the LTRs starting out, and you can see the crossovers at different places. And actually, you can use that to estimate the rate at which that kind of mutation occurs in any kind of sequence, just from what you see in this, any kind of identical sequence.
So John was also asked whether simian AIDS behaves similarly to the human HIV.
Right. It's very, in this respect, it's very similar. In fact, we also published a paper on that along with Steve Hughes and Jeff Lifson a few years ago. That as far as the properties I talked about, it's very, very similar. You get the same kinds of clonal expansion and so on.
So it's also been asked whether there are other human diseases which have some of these same properties as HIV, other than cancer.
I'm not sure.
Are there other that produce this?
There's certainly other kinds of immunodeficiencies that are caused by other virus infections in some cases, some genetically chemical certain chemical exposures and so on. Immunodeficiency is not that unusual.
Do you think there is ever always a risk that in almost any pandemic infectious disease, that the possibility could exist that a component of the infectious agent becomes integrated in a permanent sense?
Actually, I have another paper. I was lucky enough to be a collaborator on another paper, kind of a distant collaborator, that says exactly that. There are other RNA viruses and some DNA viruses have indeed ended up, bits of them have indeed ended up in our genome. We carry the first description of that was actually by a former postdoc of mine in Japan, a virus called Bornid virus, which is found in a number of mammals, a fairly pathogenic virus, but not in humans. But humans, we do carry a bit of DNA, actually a number of bits of DNA in our genome. That was put there by the machinery that moves sign elements around and line elements around, which is a kind of reverse transcriptase integrase, but it's very different very different process than what it is with retroviruses. But those can latch on to any RNA molecule and cause it to be copied and integrated. And that's how that's a very large fraction of our genome has been laid down that way, including processed pseudogenes, line elements, sign elements, and occasionally that machinery can grab a hold of an RNA virus, particularly if it has a nuclear replication phase, as like influenza virus or coronavirus.
All right. Well, I want to, on behalf of everyone, thank you very much for giving this superb talk, which has provided much food for thought in two major areas, evolution on one hand and persistence of HIV in another. So thank you again, John. I'd also like to call the attention to the fact that next week's Demystifying Medicine session is on diversity and disease in the genomic era. And the speakers are Gary Gibbons, who's director of the NHLBI, and Ann Sommer of NIDDK. So thank you all for attending, and thank you again, John.
Thank you very much for the invitation.